This is a production of Cornell University.
Wow. Thank you so much for the introduction, Michelle. I've listened to a lot of excellent seminars in this room over the years, and it's so great to have the opportunity to speak to you here today. Today, I'm going to be sharing some of my thesis research in Michelle's lab from the past few years. And what I'm going to be sharing is a tissue-specific molecular analysis of the relationship between an insect vector and the bacterial pathogen that it transmits. So the disease that I'm going to be telling you about today, if my slides advance, is citrus greening disease, which is also known as Huanglongbing. This disease originated in Asia, and it's associated with the gram-negative bacterium Candidatus Liberobacter asiaticus. And for the purposes of this talk, I'm going to be calling that CLas. And actually, I'm going to take a break to turn on my mic. Can you all hear me better now? Okay, great. Okay, so I'm going to be calling it CLas for the purposes of this talk. And you'll notice that I have to say that the disease is associated with CLas because CLas cannot be cultured. And that's also why it has to have the Candidatus designation. Now, this is the most significant disease of citrus worldwide. When trees are infected, they can remain asymptomatic for years, serving as a reservoir for the bacterium so that the bacteria can be spread between host plants within the same grove and then also to neighboring groves. When symptoms do appear, they show up on the fruit. So the fruit, they ripen unevenly and they taste salty and bitter. Ultimately, the fruit drop off the tree, and within about 5 years, this disease is completely fatal and untreatable, and the tree looks something like this. So if you were to drive through Florida, you would see groves that could only be described as citrus graveyards. So a control strategy for this disease is desperately needed. And the biological players that enable CLas to be such a successful pathogen are, of course, CLas, the bacterial pathogen, and CLas is known to infect all species of citrus. Importantly, CLas is transmitted between host plants by an insect vector called the Asian citrus psyllid, or the ACP. And interestingly, CLas does not exist in a vacuum when it's inside the insect's body. It actually has to interact with the microbiota of the insect. And the main bacterial symbionts of the insect are Wolbachia, Carcinella, and Proftella. These are housed in a specialized organ shown here that's called the bacterium. But as I'll show you in this talk, some of these bacteria are also found in other organs of the insect. Now, for this disease to be successful, these interactions between the biological players are finely tuned and co-evolved. And if we can disrupt these relationships, we can better stop transmission of CLas. In order to do that, we have to establish a better understanding of the system. So citrus greening disease was first observed in India and later China in the 1700s. So it's a very ancient disease. But it wasn't until 1998 when the ACP was first found in Florida, and then 7 years later, the bacteria was detected. In 2010, we learned that the ACP transmits CLas in a circulative manner, and that means that the bacteria actually has to circulate through the insect body. In 2011, we learned that CLas can be found in nearly every insect organ. And in 2012, sadly, CLas was detected in California, and this really underscored the importance of proper control strategies for this disease. In 2014, we learned that CLas actually replicates within the ACP, so within the insect vector. And in 2018, we learned that not every psyllid that feeds on an infected plant actually becomes infected with CLas. So CLas is not within every insect, and that means that there's some genetic heterogeneity as to the insect's ability to acquire the bacteria into its body. And therefore, if we can better understand the underlying vector capability, we can better disrupt this interaction. So these studies all together paint a picture of a bacteria that really has to interact on an intimate and co-evolved level with its insect host. And the bacteria can create an entire life cycle within the insect. So this really shows that the insect vector is an additional host for CLas and not just a conduit for it to move. In fact, when I began my research, there was a hypothesis that CLas and the ACP had a mutualistic relationship, that they were better together. This was supported by some key findings. The first is a study done by Kirsten Pell-Stalinsky that looked at the number of eggs laid by females on infected versus healthy trees. And they found that when insects are feeding on CLas infected trees, they actually lay more eggs, indicating that the bacteria is enhancing the reproductive fitness of its insect vector. And the study in our lab by Dr. John Ramsey showed that insects exposed to CLas actually show upregulation in specific enzymes in the TCA or citric acid cycle, which produces energy in the mitochondria. So this paints a picture of insects that are exposed to this bacteria that produce more energy and lay more eggs. And that's when I came onto this project. And I entered into Michelle's lab as part of a really interdisciplinary grant project. And the goal of this was to go from science to solutions for treating citrus greening disease. And we would do this by taking basic biology and more quickly translating it into field solutions that can be available to growers. So this begins on the basic biology side by doing interactome studies of the relationship between the psyllid and the bacterium. And we did this in collaboration with Carolyn Slipsky and Michael McCoss. And importantly, we had so much data from these studies, we had to properly analyze and curate this, including our genomic resources. And so we've done this in collaboration with Lucas Mueller's lab. So with all this data in mind, we would then do molecular discovery to look for molecules that can interdict or disrupt the relationship between the psyllid and the bacterium. And we've done this in collaboration with John Liss. And if any of these molecules were to work, we then still had a pipeline in place to do bioassays and delivery optimization to more quickly translate these molecules into field delivery. And we would do this via collaborations with Bob Shatters and Ed Stover. So as part of this system, we also continuously received input from scientific and stakeholder advisory boards so that we would actually be communicating with the growers about what they wanted and needed. All right, so this brings me to the 3 sections of my talk that you're going to hear today. In the first section, I'm going to be talking about more the interactome studies. And this is really the discovery basis of my research, which aims at better understanding the interactions between CLas and the ACP. And this is going to take the form of tissue-specific omics studies. In the second portion of my talk, I'll talk about the molecular discovery portion of the work, which aims to find molecules that disrupt the feeding structures of the psyllid. In the third portion of the talk, I'll discuss a collaboration with the Liss Lab that develops a method to test the binding activity of candidate molecules. So first, I'm going to begin with a discussion of tissue-specific omics. But to do this, I have to tell you more about how the process occurs when CLas moves through the insect's body. So baby psyllids, or nymphs, feed on CLas-infected trees and acquire the bacteria into their bodies, and then they molt through several different stages. And then the adult looks like this. And the bacteria moves through the gut of the adult insect and then exits into the blood or hemolymph of the insect, where it circulates. And here in the hemolymph, it can circulate until it reaches the salivary glands of the insect and can be spit into the next host plant. This interaction, as you can imagine, requires a lot of interactions between the bacteria and the cell membranes of the various insect organs. And so taking a better or more detailed look at this, I'm showing you a CT scan that I've done to show how the insect's organs are arranged within the body. And so taking a holistic view of how the bacteria has to move through these tissues, I focused on 3 that are really important for transmission to occur. The first that I've selected is the gut, because that's the first barrier to transmission. And in the first study that I'll present, I asked the question, how does the gut respond to CLas? And where does CLas go within gut cells? In the second tissue, I really wanted to look with Dr. John Ramsey at the hemolymph of the insect, because CLas has to circulate within this tissue. And in fact, it's the immune tissue of the insect. So while I don't have time to present this study, what I've done is develop a method to extract hemolymph from insects and look at the proteome response to CLas in that blood tissue. And what we found is that very specific hemolymph proteins respond to CLas. And we found that male and female insects have very different immune repertoires. And the final tissue that I'll discuss today is the feeding parts of the psyllid. So that's called a stylet. And as you can imagine, if we can disrupt feeding by the insect at this crucial first step, we can stop transmission before we get to interaction with these other tissues. And so in the third study that I'll talk about, I asked the question of whether we can identify proteins that coordinate insect feeding. So to begin, I am going to present to you a multi-OMIC study of the response of the insect gut to CLas. And to do this, I took samples of dissected guts from insects feeding on infected trees, and remember we have to call these CLas exposed because we don't know for sure if every gut has CLas, and then compared them to dissected guts from insects feeding on healthy trees. And then to these samples, I did transcriptomics to look at the RNA response and proteomics to look at the protein response. And while we made a lot of really interesting findings in this paper, I'm going to highlight a few key things. So the first thing we found was that the psylid and proteome analysis really cued us into some mitochondrial dysfunction in the gut cells of the insect. What I mean by this is, we looked back at the TCA cycle that I showed you before, and I'll remind you that at the whole body level, the insect has an upregulation of some key enzymes in this crucial enzyme cycle. And then at the gut level, when we map our data onto this cycle, we actually see the opposite effect at the gut. So you'll notice that most of these enzymes, if they're differentially expressed, are downregulated. And so this clued us into the fact that perhaps these mitochondria are undergoing dysfunction or breaking up. And so our collaborator Dasuki Amar did a follow-up study in which he took CLas plus compared to CLas minus guts and did a mitosox stain, which looks for reactive oxygen species produced by the mitochondria. And interestingly, he found that guts exposed to CLas have a much brighter staining profile. And so this piece of cell biology actually validates a hypothesis that we made using an omics data set, and confirms that mitochondrial dysfunction is occurring in the gut, and these signs are consistent with cell death in the gut. The second observation that I made from this data was that there are long non-coding RNAs, or link RNAs, in the ACP gut, and they're responsive to the presence of CLas. Now, this was really fascinating because link RNAs are really an emerging field of study and they're poorly understood. But what we do know about them is that they are regulatory transcriptional elements and they respond to stresses such as pathogen challenge and environmental stresses. And further look at our data actually showed that some of the link RNAs in the psylid contain abundant tandem repeat elements, which are derived from transposons. And this is really fascinating, and upon further literature reviewing, I found that transposable elements are really common in link RNAs, and they actually actively contribute to the evolution and function of these molecules. And when I searched our transposable elements, I actually found that they're homologous to those found and confirmed in other species. Finally, I mapped the RNA-seq reads onto the link RNA gene and found that the bulk of RNA reads actually align to the region with transposable elements, indicating that they're likely to be functional in our system as they have been found to be in others. And it's important to note that we never would have identified these molecules had we only done a proteome analysis. And then on the converse side, had we only done a transcriptome analysis, we never would have found proteins derived from bacteria within the gut. And I'm saying this because our proteome analysis was really a metaproteome, because we're able to look at the protein responses to CLas of both the insect, but also the endosymbiont proteins. And so we identified Wolbachia proteins in the gut, and that was exciting by itself because we never knew that Wolbachia was actually present in the psylid's gut. And then even more strikingly, we found that every differentially expressed Wolbachia protein was actually downregulated. This is a really striking result, and what I wanted to do was see where Wolbachia and CLas could localize within the cells of the psylid's gut. And so to do this, I did an experiment called fluorescent in situ hybridization, or FISH, which labels bacterial sequences with fluorescent molecules so that we can see where they go in their host tissues. And so this is a subsection of a psylid gut that's been stained with DAPI to label the nuclei. So this should allow you to sort of orient yourself to where the cells are located. When I use a probe against CLas, we see that CLas adopts a striated biofilm type staining pattern in the gut. And when we do the same protocol and stain Wolbachia, we see that it has a much more diffuse and punctate localization. When we merge these two images, you can see that CLas and Wolbachia actually can occupy the same cells, but they do not co-localize. So this indicates that there must be some sort of co-regulation between these two bacteria that allow them to occupy this very same cellular space, but not to overlap or compete. So this is fascinating, and to move on, I actually want to call your attention back to the image of CLas. And you'll notice that we actually see signal from CLas in the nucleus of the insect gut. And this was interesting, but it's actually not the first time that bacteria have been observed in the nuclei of their eukaryotic hosts. In fact, it's been observed that some bacteria can invade the nucleus of their host and then replicate, causing the nucleus to swell. And then the nucleus ultimately bursts and causes the cell to lyse so that bacteria can disperse more easily. And in fact, when a graduate student in the HEC lab, Marina Mann, did a nuclear study of the psyllid gut, she sees that when CLas is present, the nuclei actually do undergo swelling, and ultimately they burst completely in support of our hypothesis. But in order to really confirm that this is what's happening, we'd like to see intact bacterial cells within the nucleus. But even if we were to do an electron microscopy experiment, we actually don't have an antibody that works against CLas. So we have no way to specifically label CLas and see that the bacteria we're looking at is what we think it is. And so that brings me to my next slide, which is a new method that allows us to specifically tag proteins without the use of an antibody for use in electron microscopy. So this method takes the fish probes, which I presented to you earlier, which are made of nucleic acid, and tags the bacterial sequences. And the fish probe is conjugated with a biotin molecule. And then to that, we hybridize an antibiotin antibody containing an immunogold particle. The immunogold particle allows us to see where the antibody has gone using electron microscopy. And for validation purposes, I've employed a silver enhancement stain, which chelates silver around the gold particle to allow us to see it using regular light microscopy. When I do this, on a psyllid feeding on a healthy plant, I see virtually no staining when CLas is not present at lower and higher magnifications. And then when I do the same protocol on a CLas positive gut, I see abundant staining at low magnification, and at higher magnification, I also see that the staining adopts the same striated biofilm phenotype that we're seeing when we do fluorescent microscopy. This shows us that this method specifically tags CLas infected guts. And that's really promising. And in a preliminary transmitted electron microscopy experiment, I do see a bacterial cell of the expected shape and size of CLas that's abundantly labeled with immunogold particles, and the immunogold particles are most abundant in what we believe is the condensed DNA region. So this shows that we can specifically tag bacterial cells within eukaryotic hosts without the use of an antibody. And this is great for the study of CLas, but since these probes are so flexible, it can also be adapted to the study of other unculturable bacteria or even viruses. And from here, I want to move on to discussing the third tissue that I've mentioned to you today, and that is the stylet sheath. So in order to feed, the psyllid uses a piercing sucking stylet to pierce into plant tissue and probe until it reaches the phloem. But as it's feeding, it secretes saliva that cross-links and polymerizes to form a protective salivary sheath around its mouth parts. And you can actually see that while insects have been feeding on this cross-section of a plant, they leave behind an autofluorescent stylet sheath that shows where the insect has been probing. And you can see that actually in the plant, these sheaths undergo branching. And I'm going to talk about that a little bit more later. And then when we look at this sheath structure at higher resolution, you can see that they form a linear globular structure. Now, going into this study, we didn't know much about the sheath, but we did know that it's made of 90% carbohydrates and 10% proteins. And we also knew that it's essential for the insect to feed. So if its polymerization is inhibited, the insect simply cannot feed. We also knew that its polymerization is inhibited by EDTA, which is a molecule that binds to metal ions. So my objective for this project was to identify the proteins that are present in this structure. And when I did proteomics on this structure, strikingly, I only identified 3 abundant proteins. And they are a lacase-like multi-copper oxidase, a multi-copper oxidase, and a lacase. And I should note that we also identified a 4th unknown protein, which is metal binding, but that doesn't match to any spectra in our databases. And so this is really intriguing, and it might indicate that our genomic resources need to be improved, or perhaps this protein is heavily modified and therefore can't be identified with mass spectrometry. But for the rest of this section, I'm going to focus on the abundant proteins that we can identify, which are named MCO1, 2, and 3. So I'll note that each of these proteins has a predicted copper binding function. And therefore, my hypothesis going forward is that copper binding proteins are involved in sheath formation. The first thing I wanted to do was to see whether these proteins could directly bind copper when they're in insect cells. And so working with the Blissard lab, I expressed the MCO proteins tagged with GST in insect cells using a baculovirus expression system. I took these insect cells, And I grew them with and without copper sulfate, and then did a Western blot on the cells to see if I would see any difference in the protein banding patterns. And interestingly, what I see is that MCO1 forms a dimer only in the presence of copper. So this shows us that MCO1 at least directly binds to copper in order to form a protein complex. While I didn't see this phenotype with MCOs 2 and 3, future work could focus on co-expressing these proteins together to see whether they perhaps form a heterologous complex in the presence of copper. But this observation with MCO1 made me confident going forward in testing the effects of copper on the morphology of the sheath. And so I developed an experimental workflow that allows us to look at the effect of treatments on sheaths. And so what I do is I take insects feeding on plants and I add them to artificial diet chambers containing a particular additive that I'd like to test. And this diet is semi-solid, so I allow the insects to probe on it and they leave stylet sheaths behind. I can then collect this diet and stain it with colloidal blue. And since the sheaths are proteinaceous, they actually stain bright blue. And this assay gives us two types of information. So first, I look at the entire diet and I use image quantification to count the number of sheaths that are deposited. And this gives us a proxy measurement for feeding. The second type of information is morphological information, because I can use microscopy to look at any visible effects of the treatment on the formation or structure of the sheath. Now, when I do the first experiment and count the number of sheaths put down into diets, I see that in diets containing sucrose, compared to diets containing copper, compared to diets containing zinc, there are significantly fewer sheaths put down in diets containing zinc sulfate. And we believe that because zinc is also a divalent ion, it might be competing with copper for binding sites on these proteins and thus inhibiting sheath formation. When I do the second type of experiment and look at the morphology of the sheath, I saw something that was pretty interesting. So in a typical diet that doesn't contain any copper, I would see all linear globular sheaths that have no branches. But when copper is added, I see a greater frequency of sheaths containing multiple branching structures, so sometimes 5 and 6 branches. And I observed this and I wasn't sure what to make of it at first. And then I looked back at the picture that I showed you at the introduction of this section, and that is this. If you look at the sheaths that are deposited, what you'll see is that they undergo branching only when they're directly in the presence of phloem. And then take this together with the fact that phloem actually contains the highest concentration of copper among any plant tissue. And now my working hypothesis is that copper binding of these enzymes coordinates sheath branching only in response to the higher concentration of copper that's found in the phloem. So this is exciting, but what I really wanted to know was whether copper treatments would affect transmission of CLas into plants. And so I did the following experiment, where I took detached citrus leaves and I treated them by soaking their petioles in either water, copper, or zinc, or I did a topical swab of these metal or water treatments. I validated using ICP mass spectrometry that both of these treatment strategies successfully delivers metals into the plant tissue, but I found that petiolar delivery is the most effective. So I did both of these treatments and allowed the metal to uptake for 2 days. And then I added 10 CLas exposed insects to each leaf. And then I allowed the insects to feed for 2 days. And I chose 2 days, even though it's relatively short amount of time for the insects to feed, because I really wanted to tease out the early feeding-driven transmission events that we might see in a typical field setting and see whether our treatments would affect that. So after this feeding period, I dissected the midribs or center vein of the plants, and I collected the insects. Then I did DNA extraction and qPCR to detect the titer of CLas within these samples. And I'll note that the qPCR confirmed that all of these insects contain CLas within their bodies. And then when I did the qPCR on the midrib, the plant samples, I found that the highest level of copper treated plants actually contain the highest level of CLas within them. So this indicates to us that copper increases CLas transmission. So to review what I've shown you in this section, I've used proteomics to identify putative copper-binding proteins in the sheath. And I've confirmed that at least one of these proteins binds to copper directly. And I've also shown that sheath morphology and deposition is sensitive to copper and zinc, and that there is a branching phenotype that we see when copper is present. And finally, I show that CLas transmission is enhanced when copper is added to the plant exogenously. And this is such an important result because based on my personal communication with growers, copper sulfate-based fungicides are applied to organic and conventional citrus groves roughly every 2 weeks. So this data indicates that our management strategies as they are now may inadvertently be enhancing the psylid's ability to feed and therefore transmit CLas into plants. And so with that, I want to move to my next section. I hope this previous section convinced you that the stylet sheath is a really important structure to learn more about and ideally to inhibit. And that's what I want to focus on for this experiment. So as I mentioned to you, the goal of our big grant was to select molecules that would bind and inhibit psylid function. And the molecules that I wanted to use in collaboration with the LIST lab were RNA aptamers. And aptamers are short, small pieces of single-stranded RNA. And they have a unique sequence in its center that gives them a unique secondary structure. And the secondary structure allows them to bind to targets with affinity that's comparable to antibodies. But unlike antibodies, once the sequences are available, they can be synthesized in virtually unlimited quantities without the use of an animal, and therefore without any batch effect. Also, they can be developed by a typical molecular biologist in a wet lab. They're developed using a process called CLas, which stands for Systematic Evolution of Ligands by Exponential Enrichment. And this process requires a large library of RNA aptamers, each of which has a unique affinity for a target. And I'll note that the target is typically a purified protein in the most conventional CLas experiment. So this large library is hybridized to the target, and then washing steps remove the RNA aptamers that don't stay bound. And then those that do stay bound are eluted. And these eluted aptamers serve as the template to resynthesize this starting library. And this process is iterative, so each time it's completed, the starting library is less diverse, but has a higher affinity for the target of interest. And after the selected number of cycles, these libraries can be sequenced to identify the aptamers that are highly enriched against your target. So when Michelle and I first met, we talked about the benefits of RNA aptamers as they could be applied to agriculture. And this would be their very first application to agriculture. So what we talked about was that they're RNA molecules. So they can be delivered directly into trees via viral vectors synthesized by a collaboration that we already have in place with Dr. Bill Dawson. So Dr. Dawson takes citrus tristeza virus and deactivates it to make it asymptomatic, and he uses it as a tool to deliver RNA directly into trees. And these viral vectors are actually already approved for field application in citrus. And then alternatively, RNA can also be synthesized using modified nucleotides that increase its heat and nuclease resistance. So this type of strategy can be evaluated for topical applications. So when I began as a first-year graduate student, I wanted to try something really unconventional, which was to use whole tissues as CLEX targets instead of purified proteins. And so the tissues that I selected were whole gut cell suspensions and purified salivary sheets. And I did the CLEX experiment and sequenced the libraries, and I didn't get any aptamers that were enriched against the gut. And in light of what I showed you about the gut proteome and transcriptome, this is actually not surprising in retrospect, because now we know that this tissue is so complex. It has thousands of proteins and thousands of transcripts, and it has a really complex cell death-like response to CLas. So it's no wonder that this complex tissue was not able to enrich an RNA molecule against it. But as I've shown you, the salivary sheath is a much more homogeneous tissue with only 4 proteins abundant. And so we were able to enrich 1 aptamer against it. However, I did want more aptamers to test, so I also tried to express the MCO proteins and purify them in a more traditional CLEX strategy. And so this actually also proved to be an experiment with a lot of pitfalls, because these proteins are very large and they also prove to be toxic to bacteria. So I did work with the Blissard and Chappie labs, and we tried a lot of strategies, and we're still not giving up, but we haven't purified them yet. And so what I wanted to do was focus on the aptamer we had and just see whether it had an effect. So to do this, I first did a predicted secondary structure of the aptamer. And this is a representative structure, but I'll say that this structure has 3 potentially active stem loops. And then I took the sequence of this RNA molecule and I scrambled it to generate a control that has the same ratio of nucleotides, but a very different secondary structure. And when I deliver the scrambled aptamer control in artificial diets, the sheaths that are put down in the diets look like what you would expect. They are linear and they're globular, and there are quite a few of them. But then when I do the same experiment delivering the sheath aptamer, the sheaths that are put down are much shorter and much less abundant. And at higher magnification, I see that they also haven't fully polymerized. So first I'm showing you that there's a morphological effect from this aptamer on stylet sheaths. And then the next study I did was just to count the number of sheaths that are put down, and even counting the morphologically deformed sheaths, there are still fewer sheaths that are deposited in our aptamer containing diet compared to the scrambled control. And the next thing I wanted to know was where this aptamer can go within the psylid organs. And so I did an experiment where I fluorescently labeled both the scrambled and the sheath aptamers, and I delivered them to insects via artificial diets, in addition to a control diet which just had sucrose. So next, I dissected the heads to expose the stylet and some salivary gland material, and I also isolated the salivary glands of these insects. So this is the dissections from the control diet, and it gives us a baseline for the autofluorescence from the cuticle and eye of the insect. But you'll see that we see virtually no fluorescence in the salivary glands of this insect, and we don't see any fluorescence in what's left of the salivary tissue. When I deliver the scrambled diet, we see similarly, essentially no fluorescence in the salivary glands or in the stylet. When I deliver the sheath aptamer, I actually see that the aptamer localizes to the salivary glands of the insect. And at higher magnification, I can see that the aptamer enters into specific salivary gland cells and localizes to specific puncta, which may be vesicles or organelles. And this is an interesting result, and it potentially indicates that the activity of this aptamer may be longer lasting than its initial delivery if it can accumulate in the salivary glands of the insect. So this brings me to the next section of my talk, because here we have a candidate molecule that looks really promising, but it also presents a problem because it was selected against an entire tissue. So how do we know what it's binding? And so that brings me to my collaboration with the LIST lab, in which we developed a method. Well, first we addressed the problem of there's no method to test whether aptamers bind to their intended target specifically, which is insane because RNA aptamers are used to treat human diseases such as macular degeneration. And in this treatment protocol, they're injected into the eyeballs of human patients, but there's no method to test that they bind what we think they bind. So we took this problem, and what we would usually do in the HEC lab is an affinity purification mass spectrometry to identify the binding target of anything. And typically, affinity purification mass spectrometry takes an antibody specific to your protein of interest and pulls it down from a cell extract and immobilizes it on beads. And then this protein plus antibodies can be eluted from the beads, and then we can do gel electrophoresis or mass spectrometry to identify what came down. And so we asked the question, can we use RNA aptamers instead of antibodies to perform AP-MS experiments? And to do this, I worked with Judajit Ray, and we developed a strategy. And this is all done in human cells because human cell lines have well-characterized proteins and well-characterized aptamers to work with. So what we did was we took human cells expressing either free GFP or a fusion protein of a well-studied protein heat shock factor 1 or HSF1 with GFP, and we pulled them down using a GFP aptamer. And we're predicting that this pull down will result in meaningful interactions. So we'll identify our bait proteins as well as any interactions. And then as a control, we used a control aptamer which should not bind to any protein in human cells. And we're predicting that this interaction will identify proteins that bind to RNA non-specifically. And we've made this data available for groups who want to do affinity purification with aptamers in the future. So our experimental workflow was this. We took the GFP and the control aptamers, and we added a poly-A tail, and to that, we added a poly-T biotin. And then we used these in place of antibodies to pull down their target proteins from cell extracts that had either undergone heat shock or had not undergone heat shock. And we did that because HSF1 is known to change its protein binding activity upon heat shock. So we should be able to pick up this interaction using our method. So from this, we're expecting to pull down our target proteins, either GFP or the fusion, and then we can do a gentle biotin elution to pull down our eluates. And then to validate this method, we first did gel electrophoresis and silver staining. And when we did this, the gel that we have, well, I'm gonna break this down. So when we do this, we take non-heat shock cells that are expressing free GFP, and when we pull them down with the GFP aptamer, we actually identify the banding size of free GFP. When we do the same with a control aptamer, we don't find GFP. When we do these experiments pulling down from cells expressing the fusion protein, we do find the size of the fusion protein, but not in the control. This pattern is the same in cells that have undergone heat shock, but we see some differences in the banding patterns under heat shock, and we're expecting these to be altered interactions with HSF1 that are responsive to the stress. And then when these eluates are excited with UV, they actually fluoresce, indicating that GFP is present and that it's in its correct conformation. So to further validate this method, we had to proceed to mass spectrometry to identify what proteins were in these eluates. And the first thing we want to know is whether we pulled down our bait proteins. And so in non-heat shock cells expressing a fusion protein pulled down with GFP, we are identifying heat shock factor 1, and this measurement takes the number of spectra matching to our protein and counts them. So it's a measure of protein abundance. And the same pattern is observed when we take heat shock cells and pull them down with the GFP aptamer. And you'll note that we pulled down virtually none of this protein in our control treatments. The next thing I wanted to know was whether we had identified interacting proteins of HSF1. And so I used a tool that algorithmically scores the proteins that are identified in your sample and gives them a probability score for their interaction with your protein of interest, in our case, HSF1. And when I do this using non-heat shock cells, we identify 7 interactors. And these are predominantly published and validated interactors, which is exciting for us because we see now that we can recapitulate published results. But when I do this analysis on heat shock cells, the number of interactors jumps up to 16. And that reflects what we believe to be real biology upon heat shock. And in fact, some of these interactors have never been observed before. For example, histones have been thought to be responsive to heat shock and dependent on HSF1, but this direct interaction has never been observed before. And so this indicates to us that not only can this pull-down strategy be used to identify binders, but it can actually also probe basic protein complex biology. And finally, HSF1 is known to undergo phosphorylation, both in heat shock and non-heat shock conditions. And we wanted to determine whether we could detect phosphorylated spectra, even though we didn't do a phosphopeptide enrichment, which is typically required to identify modifications. And so at the bottom here, these are all of the phosphorylation sites that have been published for HSF1. And I'll remind you that these are cumulative and they're observed in many cell lines. And in our study, we find these sites on HSF1. And you'll notice that we identify a great proportion of previously published sites, but we also identified 2 novel phosphorylated threonine residues. And I'll end with that, but I'll remind you of what I've told you today, which is that the ACP has a tissue-specific response to CLas, and that studies at the whole organismal level often obscure information that can be really important for us to understand transmission. Next, I showed you that an RNA aptamer inhibits sheath formation. And then finally, I showed you that aptamer pulldowns can be used to identify the binding targets of RNA aptamers, but also to better understand protein biology. And that's the data that I want to show you today, but I want to take a few minutes and acknowledge the people that were really integral to the success of this work. So first, I want to acknowledge my advisor, Michelle. So Michelle is a phenomenal mentor to her graduate students, both myself and her other students in the lab. Her door is always open, and she always makes time to provide us feedback on manuscripts and talks, and she's always ready and willing to discuss experimental data and experimental design. She's also so generous about introducing me to collaborators and helping me establish productive collaborations throughout the country. And she's also wonderful to hang out with. We've had a ton of fun at meetings and on many trips. And in addition, she's someone that is ideal to talk to if your experiments aren't going how you wanted, but she's also wonderful to celebrate when you have an experimental or work success. She's also a phenomenal athlete, and she models that for her lab members. And finally, if you have not heard her karaoke voice, you are truly missing out. I also want to thank the members of the HEC Lab. So first I want to thank Stacy de Blasio. She's one of the most talented and rigorous scientists that I've ever had the pleasure of knowing. And she's also been an ally and a friend for my time in the lab. I also want to thank Mariko. So she sat 4 feet from me for 5 years, and we're still on speaking terms, but also I'm really proud to call her a friend, and her and Dave Alexander have really been a pillar of support for my time here. They even once drove 8 hours in one day when my flights were all canceled and I was stranded in Albany with 1,000 insects in tubes. I also want to thank Jenny Wilson for her friendship and her scientific rigor. I want to thank Laura and John and all the other members of the HEC Lab for hours of technical advice and friendship and moral support. I also want to thank the other mentors that I've had throughout my graduate time here. I want to thank my special committee, Gary Blissard, Josh Chappie, Bob Shatters, and John Liss. And I also want to thank Mark Fuchs for being a mentor and for making me feel welcome in Geneva. So I think I had a really unique experience with my committee because I actually got to work closely with each of my committee members and spend a lot of hours in their lab learning techniques from their talented lab members. So I want to thank Jeff Hodgson, who taught me how to use baculovirus to express proteins. I want to thank Chris Hosford, who taught me how to purify proteins. I want to thank Judah Jeet Ray, who really pioneered the biochemical aspects of aptamer pulldowns and who's just a wonderful collaborator. And I also want to thank Abdullah Ozer, who taught me everything I know about RNA aptamers and who is also a wonderful friend and collaborator. And finally, I want to thank the whole LIST lab for really making me feel welcome and for their scientific excellence and for making me a part of it. I also want to thank my family for their continual support throughout my entire educational career. A lot of them are in the audience today, so I hope that you'll have a chance to introduce yourself after this talk. And finally, I've been involved in a lot of graduate recruitment for Cornell and for our department. And the question I always get asked is, what's the best part of graduate school? And what I always know, and I always answer is, it's the friendships with the other graduate students. And so I want to thank my friends here at the graduate school for making my graduate years so fun and for being just wonderful people and wonderful scientists. I appreciate you a lot. And finally, this work was funded by the USDA NIFA and by the Citrus Research Board, and I'd be happy to take any questions.
Michelle? You want your monument?
Oh, anywhere's fine. Orlando Airport, ideally. Josh, yeah.
So I have 2 questions. Maybe I missed it. With your defects in mitochondria that you're seeing, do you know if that's because of like collapse of membrane potential and electron transport, or is it like a screwing up the dynamics of the mitochondrial network, like fission and fusion? Do you have any sense of that?
So, you know, I don't have an accurate sense of that. I will say I did the quickest experiment on the pH of guts that were exposed versus unexposed to CLas, and I found that CLas exposed guts were just a touch more acidic, and so I'm guessing that that could be a result of altered membrane transport, but I don't have a really concrete answer for that.
And then my second question is, I think I kind of asked this before, but you know, the stylet is only carbohydrate before proteins, mainly, you're arguing. So then what the hell is actually polymerizing, and how is it polymerizing, and how do you think the chemistry of these putative copper oxidases are playing into regulating?
Yeah, so my hypothesis is that the polymerization is via a process called quinone tanning, which requires oxidative cross-linking of groups particularly containing phenols. So quinone tanning has actually been co-opted by a lot of biopolymer synthesis groups because it's a really good enzymatic way to get a gel, like a biogel. And so my hypothesis is that oxidative enzymes, potentially including the MCO proteins, are physically cross-linking carbohydrates together. And I guess I would also note that since the structure is mostly carbohydrates, my hypothesis is that the aptamer is probably binding to a carbohydrate. But yeah, anyway, John.
So there's the 4th protein in the stylus that you don't know the identity of. I was wondering what the abundance of it was relative to the, of the 3.
So I would say it's the most abundant in the sheath, actually, which it's fascinating. So this protein, we don't know what it is, but it's probably the most abundant protein in the sheath, and it's also responsive to metal. So when EBTA is added, it falls apart. And it's also incredibly low molecular weight. So it's abundant, but my hypothesis is actually that for most insect species that make a sheath, typically there's something called the structural sheath protein, and one has never been identified for a psyllid species. And I did test our proteome against published structural sheath proteins from aphids, and there's no homology at all. So it's possible that this small protein might be our structural sheath protein, but further experiments might be needed to identify it.
Those structural sheath proteins, are they known to evolve rapidly?
You know, I can't answer that, but I will say that they are reasonably conserved within aphid species, but in interspecies phylogeny show that they're not well conserved between like other hemipteran insects. Oh, I'm sorry, I should be repeating the questions. I will now though, if anyone has another one. Yeah, Adam.
So I agree with Michelle, you get a mind. I was going to suggest we put you on all the orange juice cartons.
Oh.
In terms of flying after in the field, what are the potential strategies for that? If you're thinking of a foliar application, for example, do you know anything about the environmental stability of the aptamers? If you're thinking about sort of transgenic expression within citrus trees, what's the likelihood that that's going to be implemented?
Okay.
What other strategies you thought about?
Yeah, so the question from Adam is, how would RNA aptamers be delivered into trees? Okay, I think your first question was about the viral delivery. No, topical application. Okay, so the topical application, well, for certainly studies would have to be done with that, but I have, with the LIS lab, I have successfully polymerized these aptamers with fluorinated nucleotides that make them a lot more stable in the environment, but not so stable that they would stay around for years. So I think that's a more viable strategy for topical application. And we also have in the same grant network, which I talked about, we have a collaborator that works on nanoparticles to deliver small molecules. But my inclination would be to start with just modified topical applications. And then when it comes to transgenic trees, we actually have a collaboration with Dr. Ed Stover, and he's an expert in expressing transgenically molecules using a phloem-specific promoter. And my feeling in Florida especially is that if a transgenic tree reliably inhibited insect feeding or was more resistant, it would be implemented because the need is so dire. And then when it comes to viral delivery, we actually have several CTV delivery constructs in the greenhouse now. So the idea with that is that they can generate these constructs and they can be expressed in budwood that can be grafted onto a mature tree. So one potential pitfall there is that some cultivars of citrus might be resistant to CTV. So that would be a limitation of delivering that. Anyone else have a question? Jen, yeah.
So I don't really know much about insects, but I'm assuming if you inhibit the sheath formation, they starve and they die.
Yes.
So that's your target is just kill off the psyllids.
That is. Yes. So the question is, if you inhibit the stylet, do they starve and die? Is that the goal? So yes, that's the goal. So if you inhibit the stylet, they can't feed and they do starve. I'll note that they actually can go quite a while without food, but if they're not feeding, then that's solved our problem. So that's the goal, is just if we can kill all the psyllids, because they are an invasive species. It's not like we would be disrupting an ecosystem. Okay. I think those are all the questions. So thank you guys so much.
This has been a production of Cornell University, on the web at cornell.edu.